Hello everyone, it's a great honor to share our work here today. Ginseng polysaccharides prevent mastitis through Lactobacillus murinus-derived deoxycholic acid and TGR5 signaling. Mastitis is the most prevalent disease restricting the dairy industry, severely lowering milk yield and raw milk quality, impairing cow performance, and increasing farming costs. Antibiotic abuse further hinders its prevention and treatment, with global annual economic losses reaching $35 billion. Growing evidence suggests that gut microbiota interacts closely with distant organs such as the brain and liver. The concept of gut-derived mastitis has provided a new direction for revealing its pathogenesis and developing novel prevention and treatment strategies. Accordingly, LPS-induced mastitis mice showed obvious gut microbial dysbiosis and decreased alpha diversity. Fecal microbiota transplantation also reduced gut microbial alpha diversity, confirming the close link between gut microbiota and mastitis. Furthermore, FMT-LPS recipients exhibited more severe mammary inflammation and injury, impaired blood-milk barrier, and activated NF-kappa-B-NLRP3 pathway, indicating gut dysbiosis aggravates mastitis. Meanwhile, FMT-LPS also caused obvious colon and liver damage and increased multiple inflammatory markers, confirmed dysbiosis leads to systemic injury. Subsequently, we evaluated the preventive effect of ginseng polysaccharides, GP, on LPS-induced mouse mastitis. We found that GP treatment significantly alleviated mastitis, reversed gut dysbiosis, and markedly enriched Lactobacillus murinus in the gut. Next, to investigate whether ginseng polysaccharides, GP, function through the gut microbiota, we depleted the gut microbiota of mice with antibiotics. We found that the preventive effect of GP against mastitis was significantly weakened, confirming that its anti-inflammatory effect depends on the gut microbiota. Then, we used untargeted metabolomics technology to detect and analyze the fecal samples of mice in the GP group and LPS group. The results showed that GP treatment significantly reshaped the fecal metabolite profile of mice, among which the increase in deoxycholic acid content was the most significant. Through KEGG pathway enrichment analysis, we found that the bile acid metabolism pathway was significantly upregulated. Further combined correlation analysis of gut microbiota and metabolome showed that there was an extremely significant positive correlation between deoxycholic acid content and the abundance of Lactobacillus murinus. To verify the role of Lactobacillus murinus in LPS-induced mastitis, we treated model mice with this strain. It alleviated mammary injury, reduced pro-inflammatory cytokines, and increased deoxycholic acid levels. These results confirm L. murinus as the key functional strain for ginseng polysaccharides, and clarify its close correlation with deoxycholic acid. Next, we verified the preventive effect of deoxycholic acid on mastitis. In LPS-induced mastitis mice, deoxycholic acid significantly alleviated mammary inflammatory injury, inhibited the NF-kappa-B and NLRP3 pathways, and maintained the structural and functional integrity of the blood-milk barrier. To further explore the molecular mechanism by which deoxycholic acid alleviates mastitis, we performed in vitro experiments in mouse mammary epithelial cells. Deoxycholic acid activated the TGR5/cAMP-PKA signaling pathway, inhibited NF-kappa-B and NLRP3 activation, and thus reduced the production and release of downstream inflammatory cytokines. Based on the above findings, we hypothesized that ginseng polysaccharides, GP, as a postbiotic, promote the proliferation of Lactobacillus murinus. In vitro assays confirmed that GP promoted L. murinus growth, forming a synergistic symbiotic. In vivo experiments further showed that this symbiotic exerted stronger anti-inflammatory effects P or L. murinus alone in both pseudogerm-free and SPF mice. Next, fluorescence in situ hybridization, FISH, showed that ginseng polysaccharides significantly promoted the intestinal colonization of Lactobacillus murinus in mice. In summary, this study demonstrates that ginseng polysaccharides effectively alleviate LPS-induced mastitis in mice by enriching and promoting intestinal colonization of Lactobacillus murinus. Deoxycholic acid produced by L. murinus improves mammary inflammation through the TGR5/cAMP/PKA pathway. The combination ginseng polysaccharides and L. murinus as a symbiotic shows a superior protective effect against LPS-induced mastitis.